So welcome everyone. I'm glad that many of you could show up. I just wanted to say a few quick words about our guest speaker, Mike Posner, who's the inaugural speaker of this gene lecture series. We've been very fortunate in the sense that we recently got an endowment, a gift from Federico Fagin and his wife as part of their educational foundation to create this lecture series, ongoing lecture series on the neural correlates of consciousness and in general about consciousness, the topic of consciousness and awareness and things that we are actually doing a great deal of research into at the Brain Institute, the newly minted Brain Institute at Chapman. It was very important for me to bring, or at least to invite, or to have him consider, give the first lecture, and I reached out to Michael Posner from the University of Oregon at Eugene, who is a legend in the field of cognitive neuroscience and cognitive psychology and cognitive neuroscience in general. Despite the fact we had a very lousy postdoctoral student named Amiraz at some point in his career, despite that stain, he managed to survive and do quite well. And he agreed graciously, together with Sharon, his wife, to come down from Eugene and to speak to us as part of this first lecture of the Fagin series that is probably going to be ongoing here at Chapman, and I hope is going to attract people not just from Chapman but from all over. I want to tell you a few things about Mike before I hand it over to him. Mike is probably the one single most significant influence on my professional career in the sense that the science that I do today and the science that I learned how to do was mostly inspired and in many ways molded by ideas that I heard from him, not just when I was a postdoc, but over the years and before I met him by things that he has published. He's a very modest man and not a man of many words, but when he speaks very loudly and very deliberately, which is very good for the people who are listening. He's also a person who often predicts the future. I do it with magic tricks and mentalism. He does it with actually sort of figuring out what the next big thing is, which is, I think, a skill or a talent or whatever that very few people have. And I've seen him do it time and time again very naturally. Sometimes he gets it a little bit off, but usually he's right on the money. And I had a very good time training with Mike when I was younger, and to the point, we have also Jim Swanson and his wife here, and I used to, a lot of my time I was in Jim Swanson's office over in New York. It was a very delicious office, Jim. Thank you very much for letting me use it. And it's very important for you to understand that a lot of the things that you do in research in your younger days sometimes shape, your later days. So it's very important for me to introduce to you not just a mentor and a friend, but a person a great person, not just a great scientist, and somebody who, in my opinion, is one of the greatest living psychologists, American psychologist, Michael Posner.
With an emphasis on still living. I'm very glad to be here at Chapman University. I've had a remarkable day and a half. I got to see the new institute yesterday in Irvine. It's not only marked by having excellent facilities, really beautiful facilities, but also by very clever ideas, most of which, not to say anything that the last speaker was false about, did not come from me, but did come from his mind and his ability to think about how important theory and applications can be studied from psychological ideas. So this has been a very interesting aspect for me. Also, Amir is a little directive. Some of you probably are experienced with that. And he said that I must talk about consciousness, and in particular, the question of free will. Well, of course, I had to rewrite my talk because I rarely talk about these subjects. But I will talk about them as much as I can. I try to take any concept and ask what its fundamental contributions are, what are its fundamental aspects. And I believe that the problem of consciousness has two basic meanings. One of them has to do with awareness of the environment around you, as I'm aware of the faces in front of me, and you're aware of my rather loud voice. And the other has to do with control of your own activity, or voluntary control, or as it's sometimes called, will. But the use of the word will means that you could address the question of free will. But as I regard science, it's about the problem of trying to develop constraints upon freedom so that things could be predictable from past experience, from genetics, from something. So I won't really talk about free will, but I think it will be relevant to people who are working on that topic, because I'm going to talk about the mechanisms that constrain the will. That is, the mechanisms by which you instantiate your goals in actual behavior. And my general theme will be very simple. So any of you who are not psychologists or not neuroscientists and haven't got much background in this area, that's fine. I'm only going to make two major points. One of them is that we now know something about the mechanisms which allow you to control your voluntary behavior. According to most of the people in psychology and neuroscience right now, the brain has many networks that are computing different things about what you should do. So one thing is you would perhaps like to understand what I'm saying, but there are other competing things that might be computed, like the desire to leave this lecture and have a drink, start to celebrate the fact that it's Friday a little early. I know I have that feeling. So the brain computes many different possible behaviors, and somehow, in order to be coherent in your behavior, in order to be normal in your behavior, you must resolve that conflict. And so I'll talk about what we've learned about the mechanisms of resolution of conflict. And then I'll talk about something that's pretty new, but I think is hopeful, not certain about the future. And that is how to enhance that kind of self-control. That is, how to improve those networks that provide access to control. So that's the two themes. The talk will have many different methods. It will include both animal and human work. And sometimes you may get a little confused, sometimes I may get a little confused about the methods, but keep in mind the goal. The mechanisms I want to talk about, and the goal of enhancing those mechanisms. First, let me talk about the mechanisms of control. And this is a summary of about 30 years or so of work, some of it recent, much of it by other people, but which might give you a feeling that in fact we do know something. And I'm far from saying everything or everything we say is correct, but at least something about the mechanisms of control. And they are the attention systems of the human brain. And the main one that we'll be talking about today will be the one in blue here, the executive network, which includes a very important node in the anterior cingulate gyrus here in the frontal midline, in the front of the brain, right along the middle, just above the corpus callosum, the connection between the two hemispheres, lies the anterior cingulate. We'll talk about that as an important node of a network which includes the anterior insula, areas of the adjacent prefrontal cortex, and the underlying striatum, and provides an entry into voluntary behavior of the separately stored goals that you have about the current situation, whether to understand this lecture or whether to get out of here quickly. So this is one of the attention networks. There are also two others. There is a network that involves orienting to the external environment. Now it's involved competition between these slides in the visual system And my voice, my goal is to make my voice so loud that it can impose itself on the visual slides that you're seeing. And also the alerting system. You may not be aware of the alerting system right now, but midway in the lecture, particularly since it's after lunch, you'll probably feel it as your locus ceruleus gives rise to norepinephrine and tries to maintain the alert state over what might be preferred after lunch, a little nap. And we can actually measure these systems and do it very simply in a way in which both humans and animals could actually do them. And one of the ways we do this is to present you with a computer image which has a central arrow right here, central arrow, to which you press a key whether it points left or right. Now I can instruct you, if it's a mouse or a monkey, I have to train them, that the central arrow points to the direction in which they press the key, left pointer, left key, and so on. And it's surrounded by flankers which either point in the same direction, in which all the information is the same, you don't have to resolve any conflict, or point in the opposite direction in which you do have to resolve conflict, so-called flanker effect. And it takes you about 50 milliseconds to 100 milliseconds longer to respond if they're incongruent than if they're congruent. And that's a measure of your skill in resolving conflict. Probably you think that's too simple a measure to really capture. But we know from multiple questionnaires measuring effortful control in infants, of course provided by their parents, children and adults, either by teachers or by themselves, that this ability to resolve conflict as measured by this simple test correlates with questionnaire answers that parents give about their child's 3-year-old, 4-year-old, 5-year-old effortful control, ability to control their behavior and come to the dinner table when they rather would be continue to play, and so on. So there is a link between these rather precise measures of ability to resolve conflict and effortful control in everyday life. Now, before the target occurs, we can provide either a warning signal, which allows us to measure alertness, bring you to a higher state of alertness because you're expecting the target to come soon, or which tells you where the target occurs so you can shift your attention to the location of the target. These give us measures of the alerting and orienting networks, and to a large degree the three networks are uncorrelated. So if you're good at one, you may not be good at the other. We'll be talking primarily about the executive network. And it doesn't depend merely on the flanker task, the particular task we've favored to use, but this is a study by Jin Fawn in 2003 where he studied two other types of resolving of conflict. One of them, the Stroop effect, which is probably the most widely known thing in all of psychology. That is when you see the color red, you see red ink, but you see the name blue, and you're asked to name the color of ink. There's an interference effect, about 100 to 150 milliseconds. That's this one. And then there's another one that deals with the resolution of conflict between where the target occurs and where you have to respond, sometimes called the Simon effect. These three things all activate the anterior cingulate. This is their conjunction. And so regardless of how you measure with this simple task, any of these simple tasks, you can get a measure of the person's effortful control or ability to resolve conflict. And that measure then correlates with questionnaires about their child's particularly effortful control or ability to resolve conflict. Now, the anterior cingulate is a complicated system, a complicated anatomy. It's along the frontal midline, as I said before, but it stretches all the way from a very anterior part all the way to a more posterior part back here. But we can divide the anterior cingulate into two fundamental different areas. One of them involved in cognitive control. So if you do the regular Stroop effect, you get an activation along this area here. You can see all these red dots are the many tasks that have used the Stroop effect using MRI or PET to localize the conflict effect. But if I change the Stroop name from a color word to an emotional word, like cancer or death, kind of negative emotional words, or for graduate students, the name of their major professor, these will switch activation from this dorsal anterior cingulate to this more ventral part. And according to connectivity studies, the more dorsal part is connected to the frontal, prefrontal areas and parietal areas involved in cognition, whereas the more ventral part down here is connected to the amygdala and other limbic areas involved in emotion. So laid out in the anterior cingulate, we have both emotional and cognitive control. And in fact, MRI studies have shown the ability of the anterior cingulate to be connected either to the visual or parietal areas or so on, or to the limbic areas, is involved in the control of, say, the amygdala or hippocampus or parietal area. You can both increase the activation or decrease the activation. Recently, no, not that recently, but in 2012, for Weill and an all-star cast of imagers all over the world ran 725 children to adults, different ages between childhood and adulthood, on this flanker task in MRI and asked, what is it about the brain that relates to the efficiency of resolving conflict? And they found that the size of the anterior cingulate is the best predictor up until about, say, age 7. That's the age at which the flanker effect no longer improves. Reaction time improves dramatically between age 7 and adults. So you will be much faster, but the conflict effect stays about the same. Now, we thought that 7 years old, maybe 5 years old, would be about the first time that we could really measure effortful control or ability to resolve conflict in children. If you can't measure something, you usually just assume that it's not there. But fairly recently, my colleague Andrea Berger built upon a finding that was made by Karen Wynn in the 1990s. That is, she showed that infants as young as 10 months old, when showed an error, that is, instead of having a puppet put one thing in behind a screen while you're watching, then the puppet comes in and puts another thing, the screen goes up, comes down, there's only one thing present, or there are 3 things present. The infant looks reliably longer when there's an error than when it's correct. And what Berger did was to use 256 electrodes of EEG to try to localize this effect. And what she found is that that conflict in both adults and infants activates this set of frontal areas that localize to the anterior cingulate gyrus. And as shown in this next slide, in very much the same way. This 10-month-old, 7 to 10-month-old infant and the adult both show the error negativity, that is the error when the stimulus is in error, of course, they can only do very small kinds of arithmetic, like 2 plus 1 or 1 plus 1. But when it's in error, they show a strong negativity. This is the adult. It's not too striking because it's pretty simple error in the adult. The infant shows exactly the same EEG pattern, a little bit later in time, but exactly the same error negativity over exactly the same electrodes, showing that the ability to detect error, which is one of the very important aspects of the ability to control conflict, effortful control, that ability is present at least by 7 to 10 months, perhaps even earlier. And in fact, recently, people have been doing resting state MRI. That is, instead of giving the person a task, which most of the MRI studies have done, you do it while they're at rest. And of course, you can study that right down, actually, even before birth, through the walls of the mother's abdomen. And it's been shown that this node of activity in the frontal midline, in the ACC and adjoining prefrontal cortex, is present even at a few months. But it doesn't really control behavior. It's not connected to much of anything. Over the next 3 to 4 years, you can see the increase in connectivity, which allows this area to become connected to other parts of the brain and to control your behavior. For example, if you make an error, you'll slow down on the next trial, one of the most reliable things in psychology. So you're responding to stimuli, you make a mistake, maybe you say, oh, something, and you produce an error-related negativity, and on the next trial you slow down. The ability to slow down following making an error doesn't occur till 3 or 4 years later. So the infant has the node to detect error and therefore activity in the executive attention system, but it doesn't control their behavior. In this early part of life, the orienting network is much more important in controlling the behavior the infant shows, which is largely about looking time and looking away or towards something and so on. This is a study from Michael Anderson and his group using the think-no-think task. I teach you a number of associations between words. So you think of one, you have a response word to a stimulus. And then I give you an instruction. I tell you, I want you, when I turn on the red light before any stimulus item, I want you to avoid thinking about that word. If I turn on the green light, I want you to think about that word. Now, no one can do this perfectly. You do think of white elephants, as has been said when you're told not to image a white elephant. But you do reduce the activity in the hippocampus. And as you would expect from the importance of the anterior cingulate in controlling this kind of behavior, what you see here is that as the hippocampus is reduced in behavior, the frontal midline anterior cingulate is increased in behavior. And so people who increase their behavior in the anterior cingulate decrease their behavior in the hippocampus. The inhibitory effect of your effortful control, of your ability to resolve conflict on this, trying to avoid the conflict of thinking about the prohibited word. Now, on some occasions, you do think about the prohibited word. And Michael Anderson and his colleagues have shown that in that case, when you report that you thought of the prohibited word, of course, you're a little shy to do this because you know that's not what you should have done, but you're honest, so you report that you thought of it. Then you get activity here in the parietal lobe in an area that is exactly identical to the area that you get active in the orienting network when you're told to orient to a stimulus. So apparently the system builds on the orienting capacity so that if you, having been forbidden to get an item, but you spontaneously orient to it, you get activity in a network which overlaps the orienting network, at least on the right side of the brain. Okay, so this has been a brief tutorial on what we know about the mechanisms that allow you to control your behavior, voluntary control. What we know about the ability of a network, which includes the anterior cingulate, to either activate something or to deactivate or damp something down. And I think it's a surprisingly lot that we do know. And it's very relevant to exercise of free will or any other kind of goal that you have. And I do think people need to understand a little bit about this large literature that's occurred in the last several years, trying to outline this study. Of course, there are disagreements. I could be wrong on just about anything. And science is self-correcting in the sense that you may do studies that find a different story, a better story than mine. But I just think it's important to know that it isn't a blank area where we could have said, as we could have said maybe 30 years ago, well, okay, there's voluntary control, you press some key and instruction, but I have no idea what brain mechanisms might be involved. That's what I would have told you 30 years ago, too, if you were in my class. Now I'm going to try to talk about enhancing or improving this network, because, of course, as your institute represents, it's both important to do research on basic, fundamental questions of how the human brain and mind operate, but of course you'd all like to use them to better the human condition if you could do it. Maybe you can't, that's fine, but if you can, you should try. And we've been trying to see if we could enhance or improve this brain network of self-control, because we think a lot of important problems of people who have brain injury, problems of people who have addictions, problems of people who have normal process of aging. Some of their problems can be improved by enhancing this ability of self-regulation or self-control. Now, the research on this goes back to work that I did with Yi-Wan Tong in studying meditation training. He had developed a type of meditation, a type of mindfulness meditation that he called integrated body-mind meditation. The interesting thing about it, and the reason he got me involved, was he said he could get this developed in a person within 5 days that would allow a considerable amount of this kind of control. What would that allow us to do experimentally to prove that this meditation had important effects? Well, if you can do it in 5 days, you don't have to recruit a Buddhist monk and then find some person living in California who's exactly identical to that Buddhist monk but hasn't had any meditation training and compare them. You can see there could be problems in that kind of approach. Even though it could reveal important things, you could have other reasons that they might differ. But in 5 days, even an Oregon undergraduate can be recruited to spend 5 days in meditation training. And just to make sure we could rely that whatever we found with meditation training was really causal, we assigned people randomly either to meditation training or to relaxation training. Now, as you know, relaxation training, relaxing all the body parts. I can't demonstrate this for you, or I'd fall down. But relaxing all is a part of cognitive behavioral therapy. It's a reasonable way of training and plausible and so on. And so they were randomly assigned to one group or another. And we only looked at the differences between relaxation training and IBMT, or meditation training. And IBMT is, of course, Yuan's development. He's written a lot about it and has books and so on it. But it's pretty typical of mindfulness meditation, which is much more widely practiced by much more people than his particular method. But we found that in 5 days, when we compared these two groups, executive attention was better in the meditation group than in the relaxation group. The moods were better as measured by the profile of mood states. Bad moods were reduced, good moods were increased. And objective measures of stress by looking at cortisol secretion to a cognitive challenge, which was backward mental arithmetic, that produces stress. And we found that it produces less stress in someone who had had 5 days of meditation training than someone who had had 5 days of relaxation training. And this was also true of immunoreactivity, Sig-A factor, another objective measure of immunoreactivity. Moreover, the cortisol and immunoreactivity and the attention and mood improved in a dose-dependent way as we increased the meditation and control group from 5 days to 10 days to a month. That's as far as we went because of the difficulty in recruiting people, partly. The most interesting finding that we got, though, was that after a month of meditation training in comparison to relaxation training, the fractional anisotropy. Now, what does that mean? You use a measure of MRI called diffusion tensor imaging. It is thought to measure the efficiency of white matter. What it actually measures is the diffusion of water molecules in one direction, in a particular direction. If you don't have any axon tract, the water will go everywhere. If you have an axon tract, it'll follow that axon tract to some degree with error. The better that axon tract is thought, the better will be the fractional anisotropy. And what we found was that all the pathways surrounding the anterior cingulate showed better FA after 1 month of meditation training than they did after 1 month of relaxation training. To me personally, a very astounding finding which I never expected to find. And in many times after this, I had hoped we hadn't found it. Because of course, if you have an effect like this, you need to explain it. How is it? And you know scientists are a little skeptical sometimes. So when we said in our paper in 2010 that we had changed FA after a month of meditation training in comparison to relaxation, a lot of people were skeptical. I didn't necessarily pay attention to them, but these included even my brother, who's been instrumental in all my career, who's always supported me. He said, well, maybe, but not likely. So I was determined to see if I could find a cause for this effect. So I did something that we always tell graduate school students to do. We rarely do it ourselves because there's not time for faculty members, but if you've been retired like I have for 17 years, you have a lot of time. And so I read the literature trying to figure out how could this be? Most people thought then, and I think most people think now, even though scientists know better now, that white matter changes during development from infancy to adulthood, but then the oligodendrocytes, the cells that do the myelination, become dormant, and it doesn't change following that. Was the basic study. But I found that in multiple sclerosis research, people had found that once there was demyelination, the brain fought back by activating oligodendrocytes. I remember writing to the young man who had written the article and saying, gee, do you think this could happen in learning? And he said, wow, that's an interesting idea. And so I pursued that idea, that is, that maybe we could activate these oligodendrocytes by something that changed in meditation. And the most likely something was this frontal theta. Theta is an EEG rhythm, 4 to 8 cycles per second. And meditation training, many people had reported, increases the amount of frontal theta. And in fact, our own study, mostly Yuan's study, did show an increase in frontal theta following a week of meditation training, and even at rest, even when the person wasn't, as far as we knew, in a meditation state. He was just told to rest. Now, of course, what he did, we don't know. But we do see this strong frontal, increased frontal theta. And then I found in the literature, work of Gary Lynch and others, that theta could have the ability of increasing long-term potentiation and possibly changing connectivity. And so I had the elements of a kind of hypothesis, even kind of theory, about how meditation might change the brain. And I recruited a team to work on this. Actually, in fact, the leader of this team, Chris Neal, contacted me after he heard me lecture on this white matter problem and present my hypothesis, and he said, we could test that with optogenetics. We could impose a theta rhythm in the hippocampus or anterior cingulate, in the anterior cingulate of mice and see if that changed the white matter, activated the oligodendrocytes and changed the white matter. And I put this whole team because Denise Neal, also Denise Piscopo, and Aldous Wiebel were the main authors on the 2 papers that followed. What we did was to take mice who had been genetically altered so they would respond, their cells in the anterior cingulate would respond to laser light. And we implanted electrodes and lasers in the anterior cingulate of these mice. And then we imposed rhythms of 1, 8, or 40 degrees, or no rhythm. Some of them were sham operated, but never got any laser stimulation. For 20 days, just like we had done 20 days of meditation, we did 20 days of stimulation of the anterior cingulate in these mice. And since we had electrodes, we could look at the recordings, and we had mice who would increase their firing rate in a theta rhythm, and mice that would decrease their firing rate in a theta rhythm. So there's spontaneous firing, now you're increasing rhythmic firing or inhibiting the firing in a case at a 6 cycle or theta rhythm, 1, 8, or 40 cycles. 8 is the theta rhythm. Mice are a little faster in rhythms than humans. Having carried this out for 40 days, for 20 days of half an hour a day, we were then able to assess the activity or dormancy or activity of the oligodendrocytes biochemically. And we found an increase following particularly 1 cycle rhythm, not the 8 so much. Most increase was at 1 cycle per second over a control that didn't receive any rhythms, and only when they increased firing rate at 1 cycle per second, not a decrease. There was also better oligodendrocytes at 8 cycles per second, not as strong as at 1 cycle, and again, not present when we decreased. So it's the increased firing rate in the theta range or the low frequency range. We don't really know whether 1 was effective because it's very low frequency that's most effective, or whether the effective optimal frequency is somewhere between 1 and 8 hertz. It might be either one. But in general, we found an increase in active oligodendrocytes following both 1 and 8, but not for 40 and not when unstimulated. Moreover, we were able to actually look at the white matter to see if it changed. We looked at the white matter in a very high magnification, 16,000 times its normal magnification. So you could actually see the surrounding myelin. Now, the way white matter is usually assessed when you have these kind of pictures is the ratio of the internal diameter of the axon to the outer diameter, which includes both axon and myelin. Actually, it's always going to be less than 1, of course, because you have the axon diameter in both the numerator and denominator. And it's usually around 0.6. And in no stimulation, we found about a 0.72 G ratio. And it was significantly reduced following a combination of 1 and 8 cycle, both of them. We had to put them together to get a big enough N. This is hard work. And that was true in the anterior cingulate, but not in control areas like the anterior commissure. In other words, the G ratio was affected close to the area we were stimulating, but not far from it. So again, it's localization is very tight here because we know the lasers are implanted in the anterior cingulate, and we see the effects on white matter in the area surrounding the anterior cingulate, but not in other parts of the brain. Moreover, this had a relationship to behavior. Mice who are fearful stay in the dark. They cling to edges and they stay in the dark. We put these mice in a box in which they could choose either the dark area or the light area. The stronger the lower the G ratio, the more the time they spent in the light. That is, they seem to have reduced fear or anxiety by this method. Moreover, they spent more time rearing up and looking out than those that were not stimulated. So the smaller the G ratio, the more time they looked out at the outside world, the more time they spent in the light. Both of them kind of related to the fear reduction that we also found following meditation. But of course, mice aren't humans. We all know that. And this doesn't mean that everything that happens in meditation would be captured by this white matter change we have in mice, but it's a little bit hard to study all the same things that we had studied in humans in mice. So we wanted, for that reason and many others, to see if we could get this in human beings. Well, I brought you up to date. This is literature that's all been published, either by me or by others. It's all in the literature, and you can look at it at your leisure. Now I'm going to switch to things that we're doing that are not published. And I'm going to try to do this in 3 stages. The first, experiment 1, compared 3 methods of trying to establish an increased intrinsic theta in the ACC without requiring the brain to be opened and exposed. We knew the IRB would never approve such treatment of undergraduates, particularly with the high tuition they pay now. But we thought they might approve the use of small amounts of electrical activity over a set of electrodes that we knew were related to the anterior cingulate. So we proposed to study either imposed electrical activity from these set of EEG electrodes, use of auditory information, which was going at 6 hertz, and which had been reported to be effective in changing mood, or use of neurofeedback, where we informed the person of how much theta they were producing, and we told them to relax into a state where they could produce the most possible theta. That's what we did. Now, I don't claim that we had the optimal methods for doing any of this. We just did the best we could. So even a failure doesn't mean that no one, in the hands of no one, it could ever be done. But a success would mean that we'd have a way of doing it. And we did, in fact, have a success. We used 256 electrodes in an array like this. You might recognize this person. And what we found is that, finally, we also either ran the stimulation alone or together with a task that we knew activated this same area. And unsurprisingly, we used the flanker tasks, which incongruent stimuli were known to activate the anterior cingulate. What we found in this experiment, that when we used electrical activity that we thought would activate the anterior cingulate and paired it with the ant, we got the very best intrinsic theta. Now, this is from the minute after stimulation. So there's stimulation. You can't really measure how much is coming from the anterior stimulant because you're stimulating. But the minute after, you can measure it. And we measured it the minute after, and we found a rather strong increase in intrinsic theta during that minute after compared to a no stimulation group, and in fact compared to ant alone or no stimulation group. And we also found that some effect was present from the stimulation alone and from the ant alone, but stimulation plus ant was way better. That's what we found. So in this study, we used a set of electrodes over the frontal midline, which had been used to record from the anterior cingulate in many studies, and which is shown by algorithm to have its maximum effect in the anterior cingulate. But we didn't test each subject individually to get the actual structure of their brain. So these were generic electrodes. In our next experiment, having concluded that electrical stimulation was the best, by the way, we got some hints that auditory stimulation might work. It wasn't as strong, wasn't statistically significant, but something that could in fact be followed up. So in the next experiment, we took a structural MRI of each of our subjects and chose electrodes via a head model developed by our colleague Fan Liu that best activated the anterior cingulate. So we could either use the generic electrodes, which are same for everyone, very simple, or ones chosen individually for that person's brain, based on the brain. Also, you might ask the question, well, you already had meditation, why do you care? Why would you want to use theta? And of course, meditation training might be the best way. But we thought it would be great if we could have an effect anywhere in the brain. Because of course, demyelinating disorders and disorders from stroke or from closed head injury or concussion, they might occur not just around the anterior cingulate, but many other places. And so our goal was to see if we could possibly do this anywhere in the brain. Of course, we couldn't study every place in the brain. In fact, we only chose one place, the motor area, which is on the lateral frontal area, as a place to test it. It has a low intrinsic theta, and we thought it was a reasonable place to choose. So far, we've only conducted these studies using the anterior cingulate and the motor area. And we found, and of course we used both individual and generic electrodes, as I explained to you. Okay, what we found was that in fact we could get improvement in intrinsic theta, significant improvement, in both the PMC and the ACC. In the case of the ACC, stimulus plus task was the best. It wasn't as much better than stimulus alone. It wasn't that much more than stimulus alone in this study, but in the previous study it had been, and it was the best. And in the PMC, we actually found the task, which was a motor task, was the best. Task plus stimulus was about the same, and they were both better than no task and no stimulation, the control. So we could improve intrinsic theta in both the ACC and the motor area. Slightly different methods were the best, but if we used both appropriate task and stimulation, that would be as good as anything in both of these areas. And so that's what we decided to do. So the third experiment, those two experiments are done, they're written up, but they have been submitted for publication, but I can't tell you their fate yet. I'm sure they're going to get a lot of very helpful criticism. The third experiment involved 20 days of electrical stimulation plus task. That seemed to us to be the best way that we found to do this, so we proposed to the IRB and got approval to run 20 days. This is a heroic job. We have to recruit people to take 5 DTIs, I'll tell you why in a minute, 20 days of electrical stimulation. So 27, I think 27, with an intake interview, 26 sessions over 2 months. Thanks to Pascal Volcker, whose picture you saw earlier, we did complete this our first year in 7 people. So we have completed all but the last DTI in 7 people. I don't know the results, but we are proposing to run 6 to 7 more next year. We have grant money for this many. Now, I didn't have enough grant money to run a control group. So what I did was to take each person, do a DTI after they were recruited, but not start the electrical stimulation for a month and do another DTI. So DTI 1 and DTI 2 allow me to say, how stable is the DTI measure? Well, the literature tells you it's very stable, but of course, that we have to do it in our machine with our subjects and so on. So we had to measure. So in lack of a control group, we looked at that in comparison to one that occurred before stimulation and one that occurred after the 20 days of stimulation plus the task. And we've, as I say, completed that much in 7 subjects. And we plan to try to finish this next year. Now, I said 5 DTIs. We ran one a month early, one right before, then we ran one in midway. Why? Well, because our MRI data had shown something that could be very important. At 10 days of stimulation in meditation, we found a change in axonal density. You might say the inner diameter, probably, but maybe not, but that's a possibility. The MRI statistics is not exactly direct, it's a little bit removed, but we found a change in axonal density. After a month, we found a change in myelination plus axonal density. So both of them were changed by a month. So here's the idea that I have, and I don't know whether we'll ever get this result and ever be able to follow this out. But in development, first you lay down the axon tract, then you myelinate it. So in a way, this is mimicking the developmental process. And if it were, imagine instead of having to run an infant of 10 months or 7 months and comparing it with a child of 18 years or an adolescent of 18 years, waiting that 18 years. I don't really have that 18 years to wait. So instead of that, you could just look at the processes, the things that people can do before they change in white matter and after, and maybe that will give you clues or illumination to help guide your understanding of how white matter development affects the child's behavior. It's not direct, but it gives you another tool to ask a very difficult question about how the brain develops over many years of development. Although I don't have any DTI data, I do have some reaction time data from the ant. It just came in last night, so I made one more slide, and it showed a very interesting thing. Reaction time did not improve over the 20 days. Possibly having to do with how motivated you can be to run the ant over 20 days. I've done this, and I looked very much like this. You know, you get pretty disgusted running the same thing again and again. And so they did, but this I hadn't done on myself. I hadn't thought to study this, but in these people, 7 people, we got a significant improvement in the conflict measure. I find that very interesting. I don't know exactly why. We'll have 7 more next year, so we'll have 14, and we'll be able to have a result of this study. Right now, I'm very fascinated in extending this to understand the relationship between attention networks and memory networks. For example, the hippocampus. Actually, I false alarmed the word hippocampus earlier in the talk because right now we are studying these connections between the ACC and mid-prefrontal cortex, either through the thalamus. This particular nucleus is crucial according to the mouse work, or through the entorhinal cortex. And we have specific theories about how the ACC might change things, and we hope to find this. There's already a report from the literature, in fact from University of California at Irvine. I don't know if the author is in the audience or not, but these are the authors here. And they found that a very short, I think 36 minutes of theta auditory stimulation improved aspects of memory. And they did a study where you had to remember what previous classification you gave to a set of words, and they showed that memory was improved following theta. Well, the important thing is this suggests that there must be another mechanism other than the white matter change. Presumably, because it's so fast. I mean, 36 minutes, yes, you could increase theta, but no one thinks, not even us, that you could change white matter in 36 minutes. However, you could change synaptic efficacy because that occurs quickly, it's enhanced by theta, and perhaps one could get short-term effects on the synapses that would explain this memory effect. I just mention it because I know there are a lot of people looking for thesis ideas. UCI is close too. You'd have a good consultant. So think about this. Consequences. I'm getting to the end, so there's hope. You know, I don't know whether we can improve white matter anywhere in the brain or whether we can change the efficacy of synapses. I don't know yet. But I do know we've already shown there could be clinical consequences through meditation training. Yuan recruited a bunch of people to have stress reduction. Some were smokers, some were non-smokers. He then randomly assigned them either to IBMT, meditation training, or relaxation training. He found, and this is very important, that the connectivity between the ACC and the striatum, which is also part of the attention network below the ACC in the basal ganglia, the striatum, that connectivity was diminished in the smokers significantly. Now, the smokers knew nothing about smoking as far as it was related to the study. They were just recruited to do stress reduction. However, 10 days of IBMT reduced their smoking very significantly. It enhanced the ACC activity. It enhanced the lowered connectivity between the ACC and the striatum so that it was now normalized. And there was a fantastically large decrease in smoking. The red dots are before, as measured by a meter which measures carbon monoxide, and then is converted to number of cigarettes smoked within the last 24 hours. It had been pretty large before, and you can see that it's getting down to the noise of the meter here for many of these people, less than 1 cigarette. Relaxation training did nothing to smoking, but meditation reduced smoking by 60% immediately after the end of the meditation. And much of this was completely unknown to the smoker. Like they hadn't been warned that this was a study of smoking. They hadn't necessarily wanted to quit smoking. And so at the end, when they were asked about their smoking, we measured their intention to quit and their actual reduction in smoking. They were unrelated. Now, maybe this has something to do with free will, because almost every study studying smoking has selected people who intended to quit. Makes sense. But of course, I think everyone would agree if you could reduce smoking, that would be great for everyone, even those who never intended to quit. And this is the first and probably the only study, it's published 2013 in the Proceedings of the National Academy of Science, that actually studied whether the intention to quit mattered. I'm not saying it doesn't matter, but in this study it didn't matter. It's as if you changed the connectivity, you changed the desire to smoke. Now, how long does it last? We don't know. I know that question will come up. I'm going to give you a chance to ask questions, but I know that one. Oh, time for summary, 2 o'clock. Okay, free will. Your will is not free, it's constrained. It's a scientist's job to figure out how it's constrained. It's a philosopher's job to decide whether it's free will or not. I don't know. But I do know that we are on the cusp of being able to study and have already studied a lot of the mechanisms that underlie will or voluntary behavior. And we can study them in mice and in humans. Well, can we improve self-control, self-regulation? Well, I think we have a toolkit which already can improve it. Meditation training can improve it. There may be many other ways to improve it. I'm working on whether to see if stimulation by theta can improve white matter anywhere in the brain. I don't know the answer yet, but I expect this time next year I will. I said consciousness consisted of two, I thought about consciousness in terms of two underlying components, awareness and voluntary control. The mechanisms I've been talking about are really more related to control than they are to awareness. But we do know that although ambient awareness of the environment probably does not depend on the ACC, focal attention, which is kind of a voluntary act, does probably depend on the ACC. So the constraints on will may also apply to some aspects, not all aspects, of awareness. Well, thank you for your patience, and I'm happy to answer questions.